Hopefully I've got this now straightened out. It won't kick us offline again. All right, so the first couple of slides or so are just a review from really quickly to talk about what we've already covered so far. But remember, half the battle really is just knowing your enzyme class names. So if we keep going over time and time again, some of the heavy hitters. So we have kinases. And then what's the opposite of a kinase? So we have either the phosphatase, which what would the phosphatase do? It literally removes phosphate, almost always as an inorganic phosphate. And then if it transfers the phosphate without using an NTP, what is it called? Phosphorylase. Okay. And we have almost everybody's favorite to remember, which is the transferases. We have all the redox ones. What's one of the redox ones?
Oxidase.
Pardon?
Oxidase.
Oxidase. The opposite of an oxidase is a reductase. Of course, you can put the two together to make oxidoreductase. And then we have the fourth one, dehydrogenase. And the dehydrogenases are usually easy to notice because of why? NAD plus NADH, FAD, FADH2. Okay, you can't just say it removes a hydrogen because it's not really the point. A hydrogen would be a proton. Okay. And then we also have things like lyases, which you'll see once in a while. The epimerase, which what's the epimerase do? Which we'll only encounter that a couple times, or once in a while, every once in a big while.
Flips the hydroxyl on the sugar.
It what?
It flips.
Oh, it flips. That's what you said. Rips. So it doesn't rip. But right, it just goes between one epimer of a sugar to another. So it's really only something you see with carbohydrates. And of course, we have things like cyclases, nucleases, proteases. Later on, we'll talk about oxygenases and the types of oxygenase. I was trying to remember if there's something else. Ligases, technically. But it goes on and on and on. But these cover the vast majority of what we see. All right. And then, of course, we have some that are, have their specialty names, like enolase and aldolase. And we'll see some more of those later as well. Okay. Thank you. And so we've talked about the fact that the brain alone requires more than 50% of the glycogen storage on a daily basis. And some estimates have it much, much higher. I guess it depends on how much thinking you do. And so that's why between fasts, we have to have some way, I should say between meals during a fast, we have to have some way to recoup some of the sugar, okay? And so, one too many. So that's why we always have to have some form or some method to synthesize sugar since we've depleted our glycogen storage. And so that is called gluconeogenesis. Okay, and it's literally the synthesis of glucose starting from pyruvate. However, it cannot be the exact reversal of glycolysis. All right. And that was because of the 3 checkpoints in glycolysis, the 3 irreversible steps. And so, in glycolysis, to go from PEP to pyruvate and again ATP, what enzyme was that one in glycolysis? Pyruvate kinase. And then we have the fructose 6-phosphate to make fructose 1,6-bisphosphate, which was phosphofructokinase 1, or PFK1. And then finally we have glucose to glucose 6-phosphate, and that was hexokinase. And so we have to have some way to get around these. Okay, and so that's why I can almost guarantee you at some point in time on a quiz or an exam or something that you'll have to compare glucose and gluconeogenesis, glycolysis and gluconeogenesis. And so you better know those 3 checkpoints and the ways around those 3 checkpoints. All the other enzymes, the other 7 steps, are identical. Okay? So that's why they were going backwards and forwards They're all identical. It's just those three that you have to have some ways around it. And technically, there's more than three enzymes. There's more than three more enzymes in gluconeogenesis. Okay, and so make sure you know what that means. Also, where it takes place. So you're going to this semester you're going to see us do a lot of compartmentalization, and our little the happy mitochondria or whatever. We have, so there's the cell membrane. So this would be the cytosol. Oh, yeah, the cytosol. Okay, and so if this is a mitochondria, what's this outermost part called? The outer membrane. Then what's this portion called? That's the inner membrane. And then so what's this part? Of the mitochondria called? Pardon? No. It's the IMS, which stands for inner membrane space. So what's this called? Those are the cristae. And then the cristae is the little finger-like protrusions. And so what is the holy of holies in here called? What's the inside of the mitochondria called? The holy of holies.
The inner membrane space?
No, it's got a very special name. It's also the name of a really, well, I think a really horrible movie trilogy.
The matrix?
It's mitochondrial matrix.
Oh my goodness, no. I was going to say Twilight, but you know.
I've never seen Twilight. So, yeah, I thought someone was going to say Star Wars. This is mitochondrial matrix. So, within all of this, you need to figure out where's glycolysis, where's gluconeogenesis, the citric acid cycle, electron transport chain, all of those parts, urea cycle, and how it all fits in with the compartmentalization of the cell. By the way, this is not drawn to scale, okay? The mitochondria is really tiny compared to the cell, but it's just blown up, okay? And make sure that you also know why it's not the exact reversal of glycolysis, which we've already gone over, and then what the feeder pathways are. And we've already talked about some of the feeder pathways for glycolysis, and there are going to be similar ones for gluconeogenesis. All right, so this was just a table from your book that shows you the 10 steps for glycolysis, and that other color that's got the 3 that are the checkpoints, and then the fact that the other 7 are reversible, and that those are actually used in both glycolysis and in gluconeogenesis. Which unfortunately, let me put this all up. All right, whenever they updated the computers time back or so, it reversed all the animation. So this is the very first step. All right, so we have pyruvate, and notice in glycolysis we go directly from PEP or phosphoenolpyruvate to pyruvate, but we can't do that here. So it's actually a 2-step process. So if we were doing glycolysis, and this was step 10, so we have pyruvate to PEP. For gluconeogenesis, we actually have oxaloacetate, which oxaloacetate a lot of times is also abbreviated as OAA. This is a major player. Okay, there are certain common intermediates and multiple pathways. Technically, pyruvate's an intermediate not only for glycolysis and gluconeogenesis, but it's also for amino acid oxidation. Well, OAA, you're going to see it in multiple places. Okay, oxaloacetate, and it's one that you'll know the structure of by the end. Just like you should know the structure of pyruvate. Why do we know the pyruvate structure? Because what amino acid is it? Just the keto form of alanine. Well, this is a keto form of another amino acid. Okay, so this is actually 2 different enzymes. This is the first step of gluconeogenesis. Okay? And so overall, it takes an ATP and it hydrolyzes it to ADP and inorganic phosphate. But what else is it doing? What's the difference between pyruvate and oxaloacetate? What else is there? A what?
Carboxyl.
There's a carboxyl group. Okay, so it has this is a dicarboxylic acid. Technically, it's a keto dicarboxylic acid. And so that's why if you draw it out like this, it's got 4 carbons. And when we say the keto form of an amino acid, of course this is the keto that we're talking about. And so if you draw that amino acid out, what amino acid is it the keto form of? It's aspartate. I ran out of room. That's supposed to be an amino group. So that's why you already know oxaloacetate. It's just the keto acid form of the amino acid aspartate. So what class or classes of enzymes are possible for this right here? Is it really a transferase? Carboxylase. Well, it's carboxylating something. What else could it be? What other category could this fall into? Let's see, yeah, we are carboxylating. There's the carboxyl group, and it's being added. A kinase. And that is correct.
I knew it.
So would kinases generally just transfer different functional groups with ATP, or is it specifically phosphate?
It's specifically phosphate, because see, this shows you the overall, and the fact is, it's a multi-step enzyme. So it's one reaction, but it's got an intermediate there that never leaves. It's a transient intermediate of that enzyme complex. And so what they do is they just mash the two together, so to speak. And it's called, this is a carboxylase. Okay. I was jumping the gun there just a moment. And it's affected by acetyl-CoA. Where is acetyl-CoA? And why do you think it's not been on here so far. Where do we get acetyl-CoA? Make sure you know how to draw the acetyl. I don't expect you to draw the CoA part. They just always label it as S-CoA. But what's acetyl mean? What is an acetyl group?
Carbonyl with a methyl.
Right, so methyl carbonyl, and then it's going to be the sulfur, and then CoA. Okay, because that's CoAsh is a coenzyme A, and then acetyl-CoA is the acetyl group on it. But does anyone know where does acetyl-CoA come from? Where's one? We're going to find out. It's another one of these common intermediates like pyruvate and OAA. And DHAP and glyceraldehyde 3-phosphate. Some of those that we'll see over and over again. What is it the ultimate breakdown product of? It's not glycolysis directly. What other pardon? Fatty acids. It's your fats, right? So that's what's come down. So what happens here is if you have too much energy, like you get a surplus, acetyl-CoA will start to accumulate, and it's going to help kick this up, okay, which should make sense. And so the carboxylase here requires biotin. Biotin is a cofactor that we're going to see a lot of, or we'll see in multiple places, and it's really important for these carboxylase reactions. This is really annoying. Here we go. So it's a cofactor. It is covalently attached to a lysine. So this is the, my finger's a lot bigger than the thing. And here's the lysine. And remember, what's the end functional group on the lysine? In NH3 in amine, and if you remember from Organic Chem 2, those who have already taken it, remember amines can form amides pretty easy through another carboxylic acid. It's like a peptide bond. Okay, this is an amide bond, and this weird looking ring, double ring structure has the sulfur in it. Right, whoa, this is way off. Right here, that's the business group. And so these tend to help transfer carboxyl groups. We're going to see there are a couple of others that are important, but the important thing here, and I don't want you don't need to memorize the the electron pushing for biotin, but I want you to know what biotin does. Okay. And it's bicarbonate, which we have plenty of since that's a buffer system for our blood. That's the source of the carbon dioxide. I shouldn't say that. The source of the carboxyl group. Okay. And the way that it works is actually through an enolate formation. For those of you who have already had organic chem 2, remember there's the keto versus the enol form, the tautomerization, and so yeah, this never matches up quite right between the two computers. It's right here. So this is pyruvate, but this is in the enolate form, so we get the collapse, boom, and so that causes the double bond to swing out and to attack the carboxyl group that was attached here. It's not really a free carboxyl group, it's one that's attached to the biotin. And that's where we get the oxaloacetate. That's one way to get oxaloacetate. We're going to find out that there's another feeder pathway to make oxaloacetate. Actually, there's more than one, but you're going to see it a lot. All right, so now we have step 2. So this is pyruvate carboxylase. And the reason why the ATP was used in the very first reaction was the fact that it's going to activate an intermediate, okay, because the phosphate's a great leaving group. All right, and so this one, there's decarboxylation of oxaloacetate via phosphorylation of GTP, or by GTP, I should say. So what class or classes of enzymes could this possibly be? It could be a kinase. What else could it be? A what? What's that?
Decarboxylase.
Maybe a decarboxylase. This one technically in plants, and I had to look this up because the books gave different answers. Plants give it a slightly different name, and then in animals and mammals, we give it well, it makes sense once you see it. All right. And so we call it phosphoenolpyruvate carboxykinase, or it's PEP carboxykinase. Plants just call it PEP carboxylase. But to me, the carboxykinase makes sense. It both has to do with the transfer of the carboxyl group, and then the kinase is the fact that it does utilize GTP. So that's step 2, technically. Then the rest of the enzymes are the same until we get to what was step 3 in glycolysis. Right? So this is just another overall look here. So we have the bicarbonate in presence of biotin. Of course, it does require ATP. We have the pyruvate carboxylase to make OAA. And then we have this decarboxyl group, because for those, I know it's been quite a while since you've thought about beta-keto acids from Organic Chemistry 2, and we haven't covered them yet in Organic Chemistry 2. But remember, beta-keto groups are really, really important, because what happens is this is the acid, of course that's the C-alpha, there's the C-beta, and that has the keto group. And what happens is it makes this oxygen act more nucleophilic than what you would expect, because you can get collapse. This bond will be the break from the leaving group technically, so that way you get decarboxylation. You're going to, respire carbon dioxide. Then we get a boom to form the new double bond between these, but something has to leave or break, and it's just one of these bonds what breaks, and it connects to the GTP's terminal phosphate, since that's a great leaving group, and so you get GDP. You release the carbon dioxide, and you have PEP. So by everything's said and done, that's how we get around. Remember, this was like negative 61 or negative 62 kilojoules per mole, hugely energetically favorable. And so the way that we get around that is through this shunt using this oxaloacetate. All right. So now we're going to go back to Millie the happy mitochondria, or Millie the mellow mitochondria. I don't know why I call it Millie. No, I prefer Millie as my great-grandmother. All right. So I'm going to get tired of drawing this over and over and over again. I'm just going to reuse the same one. Okay, so where does glycolysis occur? Cytosol. So we have glycolysis here. Alright, yay. And so we go from glucose down to pyruvate, ultimately. And so since glycolysis occurs there, the pyruvate from glycolysis is going to be in the cytosol. And it's happy in the cytosol, it's fine, no, it's similar to alanine. Where is the beginning of gluconeogenesis? Where does it occur? Pardon? No. Good guess, though. No. There's just 2 more places left. No, there's just 1 place left. The Holy of Holies. So we have, in electrons aggregate, this is neo for gluconeogenesis. So it's in the Holy of Holies. Okay. Lots of faith integration when it comes to mitochondria. All right. And so you've got to find some way to get the pyruvate from the cytosol to the pyruvate in mitochondria. And we're going to actually find out. Now, it's not true with pyruvate, but with some of the intermediate metabolites, they can never cross. And so you have to have 2 separate pools at all times. The mitochondria has their thing, and the cytosol's got it's doing its own thing. Okay, we're going to find out a little bit about that. But there are 2 ways, ultimately, for the pyruvate to be transformed into PET. It depends on 3 things. 1 is how much pyruvate's already in the cytosol. Okay, if you've got a lot out there, then that's 1 way. Second is alanine, which why does it make sense that alanine helps dictate the amount of pyruvate you have? I mean, what is alanine with respect to pyruvate, or pyruvate with respect to alanine? Pyruvate is a keto acid form, and alanine is the amino acid form of the same compound. I mean, of the same backbone, I guess you would call it. And then we have lactate. Where does lactate come from? We have discussed lactate. It's anaerobic respiration. And all of this depends on the cytosolic need for NADH. We're going to find out NADH is one of those compounds that can never cross through the membrane. So you have to have some way to get NADH on the outside and NADH on the inside. And it's really cool, at least I think it's cool, in a nerdy biochemistry fashion, that the way that your body does this. Okay, so there are 3 methods. The first method is it's converted into oxaloacetate. But there's a problem. Oxaloacetate cannot cross through the membranes. Okay? So there's no way. So you have to have some way around that, and it's called the malate shuttle. And so because there are transporters for malate, so malate can go back and forth. Oxaloacetate can't. You need some out here. You need some in here. Okay? And so the way to do that is this first has to be transformed into malate, which I'm just going to put as mal. And likewise, so mal can then go all the way across to form oxaloacetate. The second method, we've actually already talked about, and I've even drawn this out 2 or 3 times now, could be alanine. If you have a lot of proteins to where we can't store proteins, then alanine's an important buffer, an important carrier. Okay? And so what enzyme can convert alanine, which you should by this point in time know how to draw alanine. I'm going to draw it this way, to pyruvate. This is a reversible reaction. What class of enzymes can do this? It's not an isomerase because these are 2 different groups. It's a transferase, and what transferase in particular is it? No, it's close. Wait, someone said it. It's aminotransferase. In fact, this one in particular is called alanine aminotransferase. Okay. And like I said, it is reversible. It's perfectly happy. In fact, that's important. And then finally, we have high lactate concentrations in erythrocytes or anaerobic muscle cells. And those can be converted into cytosolic pyruvate in liver cells. And then once it's that way, then it can be transported into mitochondria. Okay? And so that way, and I'll show you what, I'm going to read this off, but then I'll show you a picture of what I mean. The process anaerobically produces NADH, so therefore you won't need to use the malate shuttle. Since anaerobically you've already got the NADH, aerobically you wouldn't produce it that way, so you need the NADH. So you have to use the malate shuttle. And like I said, I'll show you what that means in just a moment. All right. Which on the one that I put online, I put boxes on this, and I was originally going to box this one too, but we're going to see this over and over and over again. Oh, let me do it this way. So let's start here in the beginning. This is the quote-unquote normal way, okay? And so if, just do normal glycolysis, we've got a lot of pyruvate, okay? So pyruvate can go into the Holy of Holies, the mitochondrial matrix. We've got pyruvate carboxylase, which is that first enzyme to make oxaloacetate. And then you just say, okay, oxaloacetate could be used, okay, in order to make the PEP. However, one way is if you need the NADH in the cytosol, the way to do that, if we don't use the malate shunt, then we don't get an NADH out there. Okay, and there are times that you need NADHs. So NADH itself can never cross the Holy of Holies, can't cross through those two membranes and the intermembrane space. So there's this cool way of going about that. There's a pool of NADH already in here, of NAD and NADH, and there's a pool on the outside. And so what happens here is oxaloacetate, oops, now it's going to sleep. Okay, oxaloacetate becomes malate. This is actually another part of another metabolic pathway. Does anyone know what other metabolic pathway that is? That has both OAA and malate? It's Krebs. It's the citric acid cycle. Okay? And so that's why we actually get multiple cycles that start to interact. And that's why that one figure from the very first chapter that I showed that had that cell and all those interlooping things, it's never just straight linear. You get interplay between the metabolic pathways. And so here, this should make sense. It's called malate dehydrogenase. It's a dehydrogenase because it utilizes NADH. It is reversible. And so it requires, and when we do the citric acid cycle, you'll be able to see what the structure of each of these looks like. It requires oxaloacetate to be reduced to malate. Malate can then go across, come back out here, and then you have the cytosolic version of it that does the reversal. So that's how you get the effective feeling of, or the overall balance between, as if you had an NADH that crossed from one side of the membrane to the other side, but it actually never crossed. It's just the electrons that did, electrons and the hydrogen, okay, because you use up an NADH on the inside, but you gain an NADH on the outside. And of course, then you just go through regular gluconeogenesis to form the PEP. The second method is in anaerobic cells. Oh, that's the wrong, sorry, I hit the wrong screen. The anaerobic cells. And so here in the muscles, or anaerobic erythrocytes, we don't have time to oxygenate, and so we just form lactic acid, and then lactate dehydrogenase forms an NADH already out here in the cytosol. So we don't need the malate shunt. So the pyruvate can just go ahead, cross right in there, form the oxaloacetate. Oxaloacetate has the carboxykinase, which can form PEP, and PEP can go back and forth. So that's why it really depends on how much NADH you need. If you need NADH in the cytosol, you've got to use a malate transporter. If you don't need it, like anaerobically, then you're fine because lactate already produces an NADH. Then the third one, which is not on here, is the fact that we can use aminotransferases to catabolize proteins, excess protein, in order to form form glycogen, which is one of the reasons why even say that someone's a diabetic, you can't just go and just eat and eat and eat, even if it's sugar-free, because you're going to find out there are ways that the fats can be stored in the form of glycogen, and now you can see how protein can also ultimately be formed for glycogen or to make glucose. And so that's why you have to be careful with, even on low carb diets, you can't just splurge and just say, okay, I'm going to eat 300 grams of protein, which don't do that anyway. But eat 300 grams of protein today, or I can go ahead and eat all this fat that I want to because these are healthy fats. Well, there's still too much of a good thing.
Like a Ron Swanson-sized steak.
Exactly. Please don't do that. All right, so the control mechanisms. There's something called the Cori cycle, which should be a review from like an A&P, I'm sure. If not, then, it's probably been a while, or even in biology. Because this is the conversion between lactate and glucose. Because you just don't want it to all accumulate, because otherwise it'll mess up your blood sugar or mess up your pH. Okay. And so, when under anaerobic conditions, as we've been stating, then you get pyruvate gets reduced down to lactate. And of course, the lactate can then be reformed through lactate dehydrogenase or be used up in lactate dehydrogenase, as we just mentioned. Okay, so the lactate from the muscle actually can be transported through your blood system to the liver, where the liver can then reoxidize it to make it ultimately go to glucose. The glucose can then be released back to the blood in order to go back and replenish the muscles. But it takes time. It's just not, immediate. So there's a cartoon coming up that shows this. And so we see this guy here. So in the muscle, under anaerobic conditions, we don't have enough time for oxygen to go through the whole citric acid cycle, so we just use lactate dehydrogenase to make lactate. The lactate then can enter the blood, go to the liver, where the liver undergoes gluconeogenesis to form the glucose, which can then re-release it to the blood in order to go and replenish the supply that the muscle has lost. So that's called the Cori cycle. All right, so we found a little bypass here. Now we've got to bypass phosphofructokinase 1. PFK1. So there's got to be a way around that in gluconeogenesis. Okay. So we have fructose 1,6-bisphosphate going to make fructose 6-phosphate and an inorganic phosphate. So what class or classes of enzymes could this be? It's a phosphatase. And so if you want to name this one specifically, what would you call it? What is it phosphatasing? Which my old boss would just cringe at that. She hated when people takes a noun and makes it into a verb. But what are you phosphatasing? The fructose 1,6-bisphosphate, right? So this enzyme is fructose 1,6-bisphosphatase. It tells you it's a phosphatase. It does fructose 1,6-bisphosphate. Okay. Now I want you to be able to use it's allosterically controlled. It's a checkpoint for gluconeogenesis. Why do you suppose fructose 1,6-bisphosphatase? Why do you suppose it makes sense that's inhibited by AMP and activated by ATP? Let's start with ATP. Why would high ATP concentrations, turn on this enzyme? Under what conditions will you have a lot of ATP? Pardon?
Respiration. Cellular respiration.
But just in general, what with respect to energy?
I use energy.
You're using energy, so you're going to have a lot of ATP? Right, if you have too much energy. So you've just eaten, everything's good. And so if you have too much energy, then you don't have to be expounding anymore, spending any more energy than you want to. You want to store it for later. So a lot of ATP will turn on gluconeogenesis. Just let me know that.
Is this step 3?
No, this is the reversal of step 3 of glycolysis, it's there's more steps in gluconeogenesis than that. So this is step 1, step 2, which brings you up to step 9 of glycolysis. Then you go from 9 to the 3, and then this is the reversal of 3. So that's why, yeah, there's a lot between here. It says that everything else that was between steps 3 and 10 on glycolysis are used with gluconeogenesis. They're shared. Okay. Why do you suppose this is inhibited by AMP? What would AMP indicate? Where do you get AMP? Where does AMP come from? What is the product of? Or one of the products of? Well, it could be from ADP or it could be from ATP. So if you've used up your ATP, AMP is one of the products of it. So that means you're low on energy. So you're going to turn off gluconeogenesis. Why does that make sense? If you're low on energy, you want to turn off gluconeogenesis. Because you don't want to be storing glucose. You need to use it to go the other way around to make more. Okay, now we have made it all the way up to the very first step of glycolysis, which was hexokinase here. But we've got to be able to go around it because it was a checkpoint for glycolysis. So this takes glucose 6-phosphate, technically nucleophilic water, and it gives off inorganic phosphate and glucose. So what class or classes of enzymes could this possibly be? That is correct. What cofactor is important when you have phosphates? And it's magnesium. Yes. So, yep, glucose 6-phosphatase. And that's the hardest one. You got to remember, this was pyruvate carboxylase, pep carboxykinase. Then these two I always thought were the easy ones because it's fructose 1,6-bisphosphatase. It really is, and this one really is glucose 6-phosphatase. Okay, and so it is activated by magnesium. But now this, I want you to start to. This I find this cool, but it's also, use a logic to be able to explain why this happens. It's only found on the lumen side, the luminal side of the ER. But only in 3 places. Okay? It's found there in your kidneys, renal cells, your liver, hepatocytes, and the epithelial cells of your small intestine. Those are the only 3 tissues that can give blood to the blood. That can give sugar to the blood. So why is that important? First of all, what happens in those 3 places? Why would it make sense that the kidney cells and small intestine would be able, what are they involved in that they're going to be able to help supply sugar to the bloodstream?
Nutrition.
Yeah, it's a type of digestion in the sense that the kidneys are filtering out, the liquids, taking care of it, and then what role does the small intestine play? It's absorption of the other materials, so. And then the liver has like 500 different things, and one of it has to be gluconeogenesis, so it should make sense for the liver there. Why is it important? So why doesn't the brain do this? Why can't the brain supply supply sugar like that, or the muscles, or stomach, or anything like that?
Doesn't have time for that.
Well, I know it can't, but why?
No, it's say can't. The brain can't expand. It's like absorb it, I guess. But the type of tissue.
Well, no, not necessarily. The large, take the colon. The large intestines are large, but it can't.
Water.
Well, no, that would explain maybe the brain, but it doesn't explain like the stomach or the heart or anything like that. Nope. I think you guys are even making it more difficult than what it really is. Well, and I guess I decided not to put it on there. Well, come on. There we go. If that enzyme was located in the other cells, then it'd make what's called a feudal cycle. That means all the cells that have glucose would make glucose 6-phosphate, and immediately they would just start to go back to glucose. Okay? So you don't want to have the enzyme that does that present in cells that's going to need the energy for the glucose. So that tissue cannot compete. The brain doesn't compete with itself for its own glucose, to make glucose to glucose 6-phosphate and immediately turn around and make it back into glucose. It would be futile. You'd be short-circuiting your battery, so to speak. Whereas the liver, we're going to find out the isoform that's in the liver and these other two specialty places, they have different characteristics for their hexokinase with respect to their glucose 6-phosphatase. Okay? So this is the reason why the glucose that's made by the liver, the kidney, or the small intestine can provide the provision for glucose for the brain, the muscles, in addition to diet. Of course, here in most Western culture, we get plenty of sugar in our diet as is, but so this is just a little cartoon to show all of this. So Lorraine, this answers your questions. The highlighted parts are the parts that are different between the two. This was from the old book, but I liked this little cartoon because it lets you show. Okay.
And you said everything else was
Right. That's why it's showing you. Let's see, because it's really tiny on my screen. It's showing from here to here. That's all identical. And it doesn't show you, but the step 2 is also identical. Well, I mean, I guess it's kind of showing you right there. And then we're going to talk about later on how glycerol can be used to make DHAP, which can then enter into. That's another one. That's why we see DHAP in multiple pathways as well. Right. So there are other ways for us to get energy. And so there are certain amino acids that are called glucogenic. There are some that are also called ketogenic because they can go on to make ketones. And the way that this is set up is you can see what amino acids are used to make or transform into pyruvate. Those that are succinyl-CoA, which succinyl-CoA, fumarate, and alpha-ketoglutarate. We're going to talk about those in greater detail in citric acid cycle. Okay, oxaloacetate is in both gluconeogenesis and citric acid cycle.
I'm guessing we don't need to memorize.
You don't. I mean, at some point, you're going to actually know several of these, but at this point in time, you should be able to explain alanine to pyruvate and aspartate to oxaloacetate. And, which, by the way, I'll just tell you the answer to this one. Glutamate and alpha-ketoglutarate, that one should be really easy. You can see it's a ketoglutarate. That's the keto acid form of glutamate. So, they call it alpha-KG, or its abbreviations a lot of times. So for those, you should be able to know, because those are the aminotransferases. All right, and so this is just... Oh, and one thing I didn't point out, but at this point in time you should have figured out. Just like in glycolysis, steps 6 through 10, you have to multiply by 2. In gluconeogenesis, the first steps until you make it to what was step 5 in glycolysis, it takes 2 of them. Like it's going to take 2 pyruvate to make 2 oxaloacetate to ultimately make 1 glucose. All right. In the last few minutes, I just briefly go over the pentose phosphate pathway because we don't have a lot of time to ever go into great detail. It's really cool. Glycolysis and gluconeogenesis are linear metabolic pathways. Citric acid cycle is going to be a cycle. This one, I don't know what to call it, a lattice. To me, it kind of reminds me of shoestrings, but you'll see the reason why. It's crisscross. It's really cool looking. But what do you suppose pentose phosphate, what is it going to deal with? What's it? Pentoses, which how many carbons are in pentose? 5, and it's a sugar. And then, of course, the phosphate. I'm just going to show all of these in that way. Oops, now go back. Okay. So this is one of the, I think we'll only see 2 or 3 times where it's in the NADP, NADPH, and that's pentose phosphate pathway. It's 5 carbon sugars, including the riboses, are made from glucose. It's important for supplying the NADPH pool that your body needs, and also for making amino acids. I'm not amino acids, nucleic acids and some fatty acids, okay? It's like the beginning to make some of the nucleic acids. And ribulose 5-phosphate, which I'm going to show you what that looks like in just a moment, it's the common intermediate. And then you'll see there's this like shuttling that occurs where it kind of looks to me like crisscrossing, where you see these 3, 4, 5, 6, and 7 carbon monosaccharides, and they should add up to 10 carbons because it would start off with 2 5-carbon containing sugars. I'll show you what I mean by that in just a moment. So this is the beginning. So first off, the way that this interacts with glycolysis and gluconeogenesis is glucose 6-phosphate. That compound is in all 3 metabolic pathways. So under certain conditions, it's going to go, glucose 6-phosphate will go through glycolysis. Other conditions, it's going to go on through gluconeogenesis and to make glycogen. And then the third condition is it's going to go on to make NADPH and the ribulose 5-phosphate, which is used for the pentose phosphate pathway. Okay. And you can see here we get an NADPH. Technically we get 2 NADPHs, and these are important for some of the anabolic pathways to make like nucleic acids and things like that. So once you have the ribulose 5-phosphate, this is why, to me, I don't know, crisscrossing. Because what happens is you start off with this 5-carbon containing sugar, and then when you have 2 of them coming together, if you notice, there's 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So there are 5 carbons in each of these, but then they swap to where now we have glyceraldehyde 3-phosphate, which has 3 carbons, and this one's got 7. And then they can swap to make erythrose, which has 4, and fructose 6-phosphate, which has 6. And another 5-carbon containing one is what's used up here to give off a glyceraldehyde 3-phosphate, which glyceraldehyde 3-phosphate and fructose 6-phosphate can then go back into glycolysis or gluconeogenesis as needed. So that's why you get this little crisscrossy shoestring looking thing. But if you notice, these all add up to 10 carbons. 5 and 5, 7 and 3, 4 and 6. Okay. Now that very last one doesn't, because it's another xylulose that comes through. And so the times that you need this, like, now I'll just quickly tell you, because usually I have you guess. The control for this is whether or not you need ATP, you need NADPH, or you need to make nucleotides. If you have plenty of energy and you don't need to make nucleotides or NADPH, it's going to go through gluconeogenesis. If you have plenty of energy but you need to make nucleotides or NADPH, it's going to go through the pentose phosphate pathway. If you don't have plenty of energy, it's going to go through glycolysis. Okay, so that's why it just depends on your energetic needs versus nucleic acid needs and NADPH needs. Okay, I'll go ahead and quickly end class with prayer. So, Heavenly Father, I want to praise you and thank you for this wonderful day, and I ask that you guide us and direct us, keep your hand over us, and help us to learn to manage our time wisely. In Jesus' name I pray. Amen.